Okay, thank you for the introduction and the invitation. It was really exciting to be invited to talk about our double haploids program. I know that a lot of people are aware of it, but maybe not totally clear on what it is or what it does. So I know that we have a diverse group in the room, and I know that not everyone is a geneticist or a breeder. And so I think my goal today is to bring as many of you along with me as I can for as long as I can. And even if you don't walk away understanding the mechanics of double haploids, hopefully you'll understand the product and also the justification for why we do it. Okay, I'd like to start off by acknowledging the OSU Barley Dream Team. We're lucky to have almost everyone on the slide here in the room with us. Scott Fisk is the assistant barley breeder. Matthew Brook is our barley postdoc who does a lot of our research work in the group. Bridget and Caleb are they kind of have their own program going with organic naked barley. And then Laura and Travis are really the ones responsible for the double haploids program. Laura on more of the greenhouse side and Travis more on the double haploids side in the lab. And then, of course, Pat Hayes, who retired, he is still kicking around every once in a while, and he's always happy to answer my emails within 10 minutes. Okay, so to start the double haploids journey, if we think about plant breeding, the goal is to combine desirable traits from multiple varieties into a single variety. And we can do that by selecting parents that have favorable traits that we would like to put together. And breeders are very good at doing this, but the problem is that it takes a long time. And so here I have kind of a schematic of a breeding pipeline, starting with the Krause, cycling through several years in the field. The main things that I'd like you to pay attention to is that these years above this orange line are mostly when we're selecting visually on the basis of traits like disease, height, maturity, things that we can more easily observe by eye. And then below the line in our yield trials, that's when we're really able to start collecting quantitative, more reliable information on traits like yield and malting quality. So altogether, this process takes around 12 years on average. There's only 9 shown here, but we often want to have potential varieties in extension and cooperative yield trials for at least 3 years. And then at the same time as that's going on, we are also putting candidates through AMBA's 2-step quality evaluation program. So it takes a really long time. And one challenge is that we can have problems arising in a time frame more quickly than 12 years. So disease can be a big one. We can have a disease emerge, and if we're not prepared for it, it can take a long time to breed a new disease-resistant variety. We actually get funding for the double haploids program from the U.S. Wheat and Barley Scab Initiative. Or we could have a new trait that all of a sudden takes off in the industry. Again, if we're not preparing for it ahead of time, it's going to take a long time to bring a variety with that trait to market. So then if the challenge becomes, how do we cut time out of that 12-year pipeline? We can look at this again and ask, okay, where can we cut time out of this process? And there's lots of creative ways to think about doing this, but looking at it more simply, okay, could we cut time out of this bottom half somehow? The challenge with that is if we compare the top half and the bottom half is really when we're getting the more reliable quantitative data on the really important traits like yield and malting quality. So we really don't want to skimp on those. As well as we might be able to predict something with genomics, we still really need to evaluate it in the field so that we're not turning something over to growers and maltsters that we don't really know what it's going to do. Okay, so if we think about cutting time out of the top half, and what's going on here anyway? Why is it that after we make a cross, we have to wait all of this time before even starting those yield trials and quality evaluations? That actually goes all the way back to Gregor Mendel, the father of genetics. So the deal is that after we make a cross, those first few generations, the genome is changing. And in Mendel's case, we see that segregation in the second generation. And it is, for breeders, this is a situation where it doesn't make sense for us to try to evaluate things like yield and malting quality that are expensive if the genome is just going to change the next generation. So how do we actually achieve that fixation in the genome? We do that through self-pollination, and barley is a naturally self-pollinating crop. So all we really have to do is put it out in the field and it will self-pollinate. And what is actually happening when we self-pollinate? The percentage of the genome that's sort of fluctuating from generation to generation gets reduced by half. So it's highest at that second generation. We have a lot of segregation, but then the more times we allow the barley to self-pollinate, the genome becomes more and more fixed. And so down here, let's say we self-pollinated to infinity. If we harvested seed from this plant and planted it again, it would be genetically identical to the parent. We can very reliably give this to growers and be able to say what it is. The problem is breeders cannot self-pollinate to infinity. So what happens practically is that many breeders, and there's a lot of variation among breeders in how they do this, but most of them will stop self-pollinating around the fourth or fifth generation and start testing for traits like yield or quality. And so if we look at that, there's not so much of the genome that is still changing from year to year. There might be a little bit that a breeder might observe, but it's unlikely to have major impacts on breeding outcomes at that point. So that's sort of the practical perspective. Okay, so we're doing this for, let's say, an average of 5 years. 5 years is still a long time. So what else can we do about that? Breeding teams working with spring crops and spring barley have creative ways of dealing with this. I used to be the breeder at Utah State, and my predecessor, David Hull, when he was breeding spring barley, he would send all the barley to New Zealand in the winter, and colleagues in New Zealand would plant it there and then send it back in time for the regular planting, spring planting in Utah. And so that was a way to get 2 generations of self-pollination in 1 year. Alternatively, breeding teams might cycle a generation in the greenhouse during the winter to achieve the same result. What about winter barley? I'm surprised how often I get the question, where's your winter nursery? Because with winter growth habit species, we really don't have an off season to take advantage of. We're planting in September and October and then harvesting in July. So there really isn't time in those just couple of months to squeeze in a whole additional generation. So doing breeding the traditional way, we still are having to self-pollinate in the field for all of those years. Okay, so that finally brings us to the question of what else can be done, and that's where we arrive at double haploids. So what double haploids are able to do is they can cut years out of this process of repeated self-pollination, such that if you make a double haploid in the lab, 100% genetic fixation is achieved immediately. So that's really the advantage of double haploids. We can take that plant and start testing for yield and malting quality because it's already genetically fixed. What is a double haploid? So plants, like animals, have sex cells, and within the barley flower, there's an ovary and there are pollen cells. And basically, humans and animals, we all have two sets of chromosomes, one from our mother and one from our father, but the sex cells at that point only have one set. And so if we go in and grab one of those sex cells, it only has one set of chromosomes. And then if we double that set of chromosomes, we can regenerate a plant from that is a double haploid. We've just made a copy of that set of chromosomes, so that's why it's no longer segregating in the genome. Double haploids is actually not that new of a technology. The first haploid plants, meaning plants with only one set of chromosomes, were observed in the 1920s. Some of the first work on trying to tissue culture haploid plantlets happened in the mid-60s. This is an image from one of those early papers. And then barley was actually, I was interested to learn it was actually an early part of the story of double haploids. So this was an early paper from 1970 in which the authors were using barley to make double haploids. Today we have, as a scientific community, we have double haploids protocols for more than 250 species. And this approach is used by many commercial breeding companies for variety development. To make a double haploid, there are two main methods to do that in barley. The first is called anther culture, and then the other option is pollination with a related species, Hordeum bulbosum. We use the anther culture method, and we follow this protocol published by Sistue, and I believe Sistue actually came to Oregon State several years ago when the lab was starting up to train the first technician. Okay, so I'm going to walk through the steps of what is actually happening in the lab. So we first grow the first generation plants. The first generation of a Krause we grow in the greenhouse, and Travis and Laura will be watching really closely, and they will collect the spikes of the flowers from the plants at a very specific stage of meiosis, when the microspores are at the mid to late uninucleate stage. Travis will then sterilize the spikes and extract the anthers. So those are the pollen producing floral organs. And he will plate around 30 anthers per spike, first on a pre-treatment medium, which will be incubated for 4 days, and then he will add the induction medium, and that will sort of trick those pollen cells to start regenerating a plant. And so after incubating for 12 to 14 days, a second round of induction medium is added. And so these are growing into mini plants, and the chromosome doubling actually happens just spontaneously. We don't have to add something to make it double. Once we have well-developed embryos, we will transfer them to our regeneration medium to try to get them to become green plantlets, and that takes around 10 to 18 days after transferring. And then at that point, we will transfer the green plantlets to magenta boxes containing kind of an agar medium, and that is to try to encourage the plantlets to grow a root system. And that's actually the point when we can vernalize our winter growth habit plants for 8 weeks. After that is done, Laura will transplant everything into cones in the greenhouse and grow those plants up to maturity. And then the seed that's harvested off of those plants is now a double haploid. So it's 100% genetically fixed and is ready to go into the field. So if we put all of those steps together, what does the year actually look like for our team? A lot of the different stages are going on concurrently as we're sort of cycling through the crosses that we do. So looking at the 2025-26 production cycle, I mentioned that we get funding from the U.S. Wheat and Barley Scab Initiative for the double haploids program. And so we do around 6 crosses per year for scab collaborators. So they will send us F1 seed from a cross, and then we will do this process for them. So currently we have 2 crosses from Virginia Tech, 2 from Nebraska, 1 from Davis, and 1 from Ohio State. And the Scab Initiative actually gets together and kind of like selects among the crosses that have been submitted by breeders to try to prioritize. And our goal for Scab is to have 1,000 fertile double haploid plants every cycle. And then the barley breeding program at Oregon State is completely run off of double haploids. We have 5 crosses going through this cycle, but in the past, I think it was like 5 to 7 per year. And so altogether, our process for a double haploid production from a cross is around 1.5 years. But you also need to consider that that is calculated starting from the F1 seeds. And so you still have to think about how much time it takes to make the initial cross. Okay, common pitfalls that I want to touch on, things that we run into that are challenges for us. In the Sistué protocol, he reported that this protocol has a high rate of spontaneous chromosome doubling, but we can get full plants where the chromosomes never doubled. So they're haploid, and because of that, they're sterile and they look a little bit different. They have a high number of tillers, really narrow leaves. And then at the same time, we can also get tetraploids, so ones that have doubled twice, and those will have abnormal seeds, fewer tillers, and thicker leaves. So those plants we just have to discard. Another common pitfall is albino plants. The frequency of albinos that we get is highly dependent on the genotype of the barley that we're working with. So a couple of different authors have explored this and found that there's a ton of variation in barley genetic diversity for the production of albinos, and the mechanisms for this inability to produce chloroplasts are not well understood. So we do get albinos, but I would say albinos, tetraploids, and haploids are things that we can, for the most part, deal with just by increasing our numbers. The problem that is maybe the biggest for us is recalcitrance, which we kind of heard about in the context of gene editing as well. We have found through the Scab Initiative that there is considerable variation within U.S. barley germplasm in terms of recalcitrance to the double haploids protocol. And in our experience, we have found that 2-row winter types work best, and we have the most problems with 6-row and or spring types. They're more likely to be recalcitrant. So we have a working hypothesis about what's going on here, which is that Double haploid technology has been used in European barley breeding programs for many years. And as a result, those breeding programs would have naturally selected material that does well in the double haploids process. At the same time, the OSU barley breeding program started doing double haploids in 2012. So we have also been selecting for a material that does well. So for the most part, we don't have recalcitrance issues when we do our own OSU crosses. We usually have the most problems with recalcitrance with certain crosses that we get through scab. And so our working hypothesis is that germplasm containing European and or OSU material in the pedigree is less likely to be recalcitrant. I know that's kind of a dissatisfying answer. I wish that we could replicate 30 years of selection in the time frame of a 1-year scab grant, but we're always brainstorming about ways to try to improve this issue for especially those 6-row and spring type crosses. Okay, so a real world example of variety development with double haploids. So the OSU barley breeding program released Top Shelf recently. This is a 2-row GN0 winter barley where the original cross was made in 2015. The double haploids were produced in the next couple of years. One challenge with double haploids, I mean, this is kind of the same with traditional breeding methods too. Like the amount of seed that comes off of a single plant isn't enormous. And so we can't, in that very first year, go to highly replicated trials all over the region. So we do go through a year of single row or mini plots in the field. We can start to collect some preliminary data on those. And then we can go into yield trials, cooperator trials, the AMBA pilot scale program. And then we released Top Shelf in 2023. So that was kind of the time frame, 2015 to 2023. We figure we're cutting off possibly 2 to 3 years from the whole process, depending on how the breeder is setting up their program. So kind of bringing all of this together, the advantages of double haploids is that it's time saving. We can develop varieties and release them faster. And we also have that full genetic fixation right away. So we're not losing material because it's still segregating in the field. I touched on some of the efficacy issues, so that's maybe the drawback of the approach. And then also there's a high cost to this. So you might imagine that this requires a number of greenhouse and wet lab facilities. It also requires skilled personnel. We just went through a personnel change with the double haploids. Wet lab person, Travis, is still relatively new, and it's finicky. So I think there's a learning curve to it. And I think the high cost aspect of it makes it kind of cost prohibitive for a lot of public breeding programs in the US. But commercial breeding programs, especially in Europe, are using double haploids. And then a lot of the commercial corn breeding programs in the US are also using double haploids. And related to that, there are a number of barley varieties that are with us today that were developed through double haploids. I just dug through the USDA's PVP database and was like opening up each one of those PVP documents, and in there was able to find sort of the breeding history of these different varieties. And they say right in there, DHs were produced during this year. And so these are definitely not the complete list of barley varieties developed with double haploids, but a number of them that are with us today are from double haploids. So I think, in conclusion, I think it's a really important technology. I think that even though it does have some major drawbacks, I think having it accessible as a resource is really important in case we do have major issues that arise that we need to respond to really quickly. And I believe that the OSU double haploids lab might be the only public double haploids Barley program in the US that's offering that. So I think maintaining that sort of skill set in the community is really important. Okay, I'd just like to acknowledge the OSU Barley group, the ARS colleagues that we work with, all of the WMBT collaborators and SCAB collaborators, and our funding comes from AMBA, SCAB, USDA, BPI, SGGI, and formerly the Wheat Commission and Great Western Malting. Thank you so much for this opportunity to speak with you, and let's work together and go bees.
Margaret, we have time for a few questions. If anyone has any questions, I'll swing over to you.
Okay, thanks. Great talk. And I just want to testify that your protocol works for wild barley as well, from my experience. So Pat made a double haploid for us in Spontaneum, and it was brilliant. It worked really nice. But what I wanted to ask is, my name is Eyal Friedman. I'm from ARO, Vulcani, Israel. And I wanted to ask if you noticed any selection during the process in the population for certain regions in the genome. Whether you see allelic differences that are translated into different penetrance in the population.
We have not looked at that recently, as far as I'm aware. I believe some papers have looked at sort of like segregation distortion and things like that. If we were to look at it, I think that we would see like alleles that are common in the Oregon State program are the ones that, show up in the successful double haploids. One thing kind of in that direction that we have thought about doing is like, with the recalcitrance problem, with the mid to late uninucleate stage, we currently do not take every genotype and look at it under the microscope. We are using more of like a phenological marker to get at when that timing is happening, but there is a decent chance that there is variation for the relationship between that phenology and the mid to late uninucleate stage. So I think that's something that we could explore more to try to improve our protocol and, not just be selecting for this one thing, if that makes sense.
I can tell you from our experience that there is distortion. I mean, very clear in that naked double haploids, and you see it with wild. You see certain distortion in certain parts of the genome, which, it's interesting to realize whether there is any genetic control.
Yeah, it's definitely something that should be explored more. It's just hard to do it and do the production scheme at the same time.
I'm just a little curious. Can you change the time frame of that? Is like, so the problem with 1.5 years is it ends up actually really being 2, right? Because then with a winter crop, let's say you get it done in spring, we're not going to plant it till the fall anyway. Whereas if you could shave 4 or 5 months off of that, you might be able to do like some late planting or something like that in a single rows. Maybe even ideally get it in a normal planting time. Is that 1.5 years like almost impossible to get around? Like, could it be reduced to 1?
I would probably defer to Laura Helgerson on that question. But I mean, I guess you could think about like, could you take the seed and like put it in a speed breeding context and like maybe you could take some of the time off.
I guess where I'm coming from is at that point where I would be in the same stage on like a population would be in an I'd be going into F4s. So I'd only be 1 year behind, but instead of having 1 or 2 or 5 or 7 populations, I have hundreds.
Right.
And they've been under selection in the field, right? So like, I'm just curious to know, if you could drop that down by just a few months, you might actually really gain that 2 years as opposed to 1.
Yeah, I think it's a great point, and I think there probably are ways to achieve it, but so much of it depends on the capital and the human capital that you have as well.
Yeah.
Okay.
Okay, out of the interest of time, to keep us on schedule for lunch, we're going to move on to the next speaker. So if you have any questions for Margaret, I'm sure you'll be here the rest of the day. You're not running away. So just hunt Margaret down and pester her with any of your questions. Sorry.